everyone, my name is Julie Jung, and I'm a postdoc at the University of Utah in Salt Lake City, and today I'm going to share some exciting results, exciting discoveries that we made in our neighboring Great Salt Lake. For some context, the shrinking of saline lakes is a growing global problem. Large saline lakes like the Great Salt Lake represent almost half of the volume and a quarter of the area of all lakes globally, but many are in dramatic decline. Utah's Great Salt Lake is no exception. What once was the largest saline lake in the hemisphere is rapidly disappearing. Now, why does this matter? Well, saline lakes are critically important for wildlife, for all sorts of industries, and for human health. This lake serves as a migratory hub for more than 10 million birds, and it generates billions of dollars annually through industries such as mineral harvesting and brine shrimp harvesting. Lastly, it's a terminal lake in a heavily mined region, so that means that any exposed lake bed contains toxic wastes that can get swept up by the wind and end up in our lungs. So I don't have time much to talk about this today, but if you're interested, please stay tuned because we have a paper in review right now which looks at various harmful toxins sequestered in this lake and in its lake bed and the risk involved in letting it dry up as much as it already has. Okay, so this satellite image shows the lake's levels in 2022, surrounded by miles of dried up lake bed. And I live right there in that white box, so I am especially worried about this. And if you take a walk alongside the water's receding edge, this crisis is quite apparent. We start to see these reef-like structures emerge from the water and bleach in the sun. And these bacteria-built microbialites were the primary form of life on Earth for most of Earth's history. But modern occurrences of these mounds are really rare. And the Great Salt Lake is now home to the world's largest assemblage of microbialites in the world. It spans about 20% of the lake's bottom, but only in these shallow marginal ramp areas, which are highlighted in red. And these microbial mats are the primary habitat for brine fly larvae, which in turn are the primary diet for many of the bird species at this lake. So basically, this entire ecosystem depends on the health of these very vulnerable reef-like structures. Now, before I started my postdoc, there were reportedly only 2 animal species thought to inhabit the waters of the Great Salt Lake. That's brine shrimp and brine fly larvae. But being a nematode lab, we knew that nematodes are the most abundant and widespread and resilient animal phylum on this earth. They make up about 80% of all animal life by number, so 4 out of 5, all animals, is a nematode. And they're found on every continent and all ecosystems. So for example, in Lake Mono, a saline soda lake in California, miles under the Earth's surface in a South African gold mine, and in some of the coldest and driest soils on Earth. So when considering what microfauna could possibly inhabit the super salty conditions of our Great Salt Lake, we predicted that maybe if we're lucky, we'll be able to find some nematodes. So to do this, we went in search of nematodes in, on, and around the Great Salt Lake, basically by any means necessary, so by bike, ATV, hiking, stand-up paddleboard, like everything but horse, basically. We went in search of nematodes. We did this in 6 different sites with a range of salinities during different seasons in 2021. This was during a really historic low in lake elevation. And these are our sampling sites. The first 2 sites are basically fresh water along the Weber River. That's one of the inputs into the lake. The third was brackish, and then our last 3 sites were all well within the hypersaline range. So this was sometimes 4 or 5 or 6 times as salty as the ocean. And we managed to find nematodes in every site. So on the y-axis, we see nematode abundance. Per handful of soil, it's 100 grams on a log scale. And these nematodes can live happily in Great Salt Lake water for quite a long time. So time here is on a scale of days to weeks to months, not seconds, not minutes. But they die pretty much immediately, well within 5 minutes if they're placed on standard NGM plates, compared to freshwater worms, which do fine. Conversely, we took 2 common laboratory nematodes, and we put them in water from the Great Salt Lake to see how many of them would survive. They both didn't do so great either, all dying within 5 minutes. So perhaps not surprisingly, these nematodes are highly adapted to their respective environments. So to summarize, we discovered some novel nematodes in the saltiest environment that we've ever found nematodes in, and it turns out they need salt to survive. Next, what kind of nematodes do we have here? So to get a sense of the nematode diversity across our sites, we Sanger sequenced hundreds of worms. Much of this sequencing was done by some very talented first-year undergrads in a genetics class that I taught as part of the Science Research Initiative at the University of Utah. The details of this tree are not super important, but what's really cool is that our sampling recovered 2 out of the 3 major lineages that exist within nematodes. And for the most part, our trees recapitulate the established global phylogeny of nematodes just by sequencing a few select loci. Simply told, each little block here represents a single nematode colored by family, and we found that diversity drastically decreases with increased salinity. So nematodes found in our freshwater and mostly freshwater sites belong to a bunch of families, whereas our saltwater sites by and large only turn up one family, and that's the family Monosteridae. So if we take that big phylogeny from before and we zoom in on just this family Monosteridae, we seem to have at least 2 fairly well-defined clades that separate really nicely based on their ecological differences. So our salty worms all seem to be in the genus Diplomoides. So along with Thomas, who is an undergrad in the lab, we recently confirmed using various types of microscopes that these nematodes morphologically resemble other Diplomoides nematodes, with some features, though, that set it apart from any other known species in this genus. So we are currently, Thomas and I, are currently working up a manuscript to describe this species morphologically, place it within known phylogeny, and then present the first chromosome scale genome of a monosterid nematode. Interestingly, we also did see some morphological differences that are unresolved from 18S sequencing. So it makes us think that we probably have multiple species living in this lake. And this is important because the species diversity within this family from these sites gives us a sense of how long these nematodes have been here in the lake. So if we had found that all the monosterid nematodes were a single species, that might suggest that the nematodes came relatively recently, so perhaps on the shoes or the clothing of some visitors to the park. But there seems to be some species diversity here within this family. And so the most parsimonious explanation in my mind is that they've been there a really long time, and just the natural mutational clock has caused them to diverge from each other and adapt. Okay. So monosterids are found all over the world, and they demonstrate this unique capacity to adapt to extreme environments. For example, they're one of the most abundant animal taxa in deep-sea hydrothermal vents in the abyssal zone of the ocean. We still have no idea, though, what allows them to live in these places where very little else can live. So with the help of a lot of collaborators sending in soil and water samples from all over the world, including some from these deep-sea environments, we are currently undertaking global sampling and whole genome sequencing for members of this family. Okay, so next, how do these nematodes survive the extreme salinity in this lake? We thought that maybe the lake's microbialites would provide some sort of refugia for the nematodes. So to find out if these worms prefer to live in these mounds, we sampled either directly from the microbialites or from the sand between microbialites, but right next to the microbialites, or from a mix of the two. And this, we found far more nematodes in the microbialites than from the mixed samples and from the sand samples. So what is driving this preference? We could tell from the mouth morphology and just the behavior of these worms that these guys are bacteriivores. And we hypothesized that the bacteria in the microbialites could be playing a role here. So we did the same simple survival assay as before with our laboratory nematodes, but we filled in these middle values between with the dilution series of 0 to 100% Great Salt Lake water. And when I say we, I mean myself with the help of Maxime, my undergraduate co-author. He did not respond in time to my text to send a really nice photo of himself, so this is what I could find online. But both of these nematodes typically eat E. coli strain OP50 in the lab, so that was our control shown here. We also made 4 cultures of microbialite bacteria, and we reared nematodes on them to adulthood. So miraculously, microbialite microbes improved nematode survival in all 4 cases here. So this was really exciting for us. So far, all of these cultures, though, are mixed populations. So next, we wanted to get a sense of what specific bacteria could be beneficial for these nematodes. So we sequenced nematode-associated microbes. And the diversity of microbiota here in our samples were pretty consistent across individuals. They're all dominated by these 3 bacterial phyla, and these are pretty commonly prevalent in hypersaline waters all over the world. Interestingly, though, there is considerable overlap between bacteria associated with nematodes and those associated with microbialites. So these 8 bacterial orders shown in textiles are those ones that kind of cross that Venn diagram. And stay tuned because we have some really cool projects planned that were recently NIH funded, and they're on the horizon. They involve isolating these bacteria and sequencing their genomes and then cloning them into E. coli to make sort of like a microbe medicine for enhanced survival in laboratory nematodes. So in summary, the microbialites that these nematodes live in could have played a role in helping them adapt to harsh conditions. Microbialite diet enhances nematode survival, and we're currently trying to culture and describe not only these nematodes, but also their bacterial symbionts. Okay. This research holds more and more importance as we explore other worlds in our solar system and find evidence of conditions that might support extremophiles. The icy moons of Jupiter and Saturn hold these really vast subsurface salty oceans. Pictured here, Mars exploration rovers have revealed mineral deposits strikingly like salts found in extinct lake beds in the Atacama Desert or Death Valley. And research at analog sites like those and like the Great Salt Lake has really important implications for where complex life might be found elsewhere. Okay, a shameless plug, if I have the time, which I believe I do, I am tiptoeing my way in towards entering the faculty job market. Ultimately, I aim to develop a life that takes a lab that takes an integrative look at how stress, generally, as an environmental factor, affects plastic phenotypes. I have not always studied nematodes, so in grad school I studied a type of behavioral plasticity in tropical tree frogs, where embryos will hatch early as a result of hypoxia or different stressors, so hypoxia, parental neglect, or predator vibrations. So I hope to continue this work in glass frogs and red-eyed tree frogs specifically. Finally, this salinity stress stuff that I presented today actually started out as a pandemic side project when I wasn't allowed to go into lab when I started my postdoc. The majority of my postdoc work since then has been on dietary stress in diplogastrid nematodes, and how that kind of serves as an environmental cue for these really cool divergent mouth morphologies. There's been a couple talks during this conference that have been about this. And if you want to listen to more, though, I'll be presenting some of our recent transcriptome data as a poster later today, if that interests you. Also, if you live next to a cool saline lake that we should check out, or if you just want to chat about future collaborations, you can find me there later today. So I want to give a huge thank you. Sorry, one more slide. I want to give a huge thank you to the Werner Lab and others in the department for giving me a scientific home. Thank you. I do have time for some questions. Yeah.
Did you find any other biofauna in these samples? So expect to find the odd rotifer?
Yeah. Yep. Diatoms, rotifers. I was really excited by some rotifer talks that I went to today and earlier in the conference. We could definitely, if anyone studies rotifers, I don't know anything about them, and I'd love to learn more. Yeah.
I'm sorry if I missed this, but do you know anything about the reproductive mode of the rotifers? Like, are they alpha-gate outcrossers themselves?
From our SEN images, I haven't been able to find a male. I think they're really rare if they're present. I do think we saw some egg-like structures in like an extracellular membrane around some of the worms, so I think that membrane is really important. So I think we're trying to culture them. I think culturing them with microbialitis is important for that. Thank you.
Cool. Okay. So my name is Wooldridge. I'm a postdoc from UC Santa Cruz. Today I'm going to be telling you about some recently published work I'm really excited about studying black abalone, and at the end I'll touch a little bit on future directions as well. So depending on where in the world you're from, you may be or may not be familiar with abalone. They're these large, actually quite long-lived gastropods, just big old snails with this really beautiful morphological diversity found in all sorts of environments, subtidal to intertidal. And they are often important components of marine ecosystems. In the case of black abalone, I'm showing a toy kelp forest California ecosystem here. They're important grazers and prey items as well. And abalone have been important to humans for a very long time as food items. The meaty, kind of fleshy part there, common part of all sorts of dishes for humans across millennia, and as a result, this has often resulted in unsustainable fisheries. A little bit more on that later. But it's not just the meaty foot. The shell has also been prized by humans because of the beautiful iridescence it has, particularly once you polish it. And shells and shell fragments have been used as jewelry, currency, arts, you name it. And today I'll be focusing on this one species in particular in all its glory here, the black abalone, which is a bit of a California icon, distributed, let's see. That range map is supposed to be filled in, but it's a west coast of North America species through California and down into Baja California. All right. And the black abalone has experienced a whole bunch of threats, some of which are common to many marine species, and you're probably familiar with them. Classic things like over-harvesting, which I alluded to. At the height of the black abalone fishery in particular, you're talking about tons being pulled in annually, just in a way that was not sustainable. And of course, other classic threats like habitat loss or pollution is removing the intertidal habitat in which these black abalone sit. And even though sea otters are cute, here's an argument to not like them. They are primary predators of black abalone. They can actually have like a pretty substantial effect on abundance through time. And then one particular threat that I'm going to focus on for a quick second is this disease known as withering syndrome. And this is actually a recent thing. It's a bacterially caused disease that infects the gastrointestinal tract of black abalone, essentially making it so that they can't digest food and they just wither up and starve. And it first appeared in 1985 in the California Channel Islands and spread quite quickly after that. In many places, particularly in Southern California, so south of Point Conception, resulting in local extirpation, severe decline. It was also occurred in the north, but to a lesser degree. And it's primarily this disease on top of a long history of fishing that resulted in the closure of this fishery a while back, actually now in '93, and then eventually this being listed under the U.S. Endangered Species Act in 2009. So given that abalone are important to us as humans and also important to coastal ecosystems and just cool intrinsically, it begs the question, what are the recovery prospects for this species? And I'm going to be talking about one particular angle on this today, as you can guess by the talk. It's a conservation genomics angle and what that can tell us about the future of this species. And these are critically endangered, so you can imagine sampling is difficult. And in the Shapiro lab at UC Santa Cruz, they developed a really cool method, which is essentially an abalone COVID swab, where you take a nylon tip swab and you go in and get the mucousy part, and from that you can get high quality whole genome coverage, and you can also get some other things too, which I will touch on later. But thanks to that, we were able to get around 130 samples distributed across mostly the California range, a little bit into Mexico, and from a bunch of different sites. So with this sort of data, you can answer a bunch of different questions. Some of the more obvious ones to start to look at are things like genetic variation. Given this huge bottleneck in recent history, what genetic variation is there? And the long and short of it is that there is a lot of genetic variation if you want to put it in terms of diversity within a given site, how many sequence differences there are between individuals, say, in one of these Channel Islands. It's quite high, and higher than a lot of other broadcast spawners and most vertebrate species of similar concern with that endangered status. But I think the more important take home here is actually that this level of diversity shows no significant association with where we sampled from. And again, this bottleneck that happened in the 80s was much more severe in the South, which is kind of interesting. A more explicit demographic inference in very recent time, interestingly, does show a real severe bottleneck of about the magnitude ecologists reported. Around 15 generations back, I'm a little hesitant. It's tempting to speculate that's the disease thing and then there's recovery. Just a little hesitant because generation time is complicated in abalone, hard to pin this to real time. But what I can say is that bottleneck and the recovery seem to be robust to a lot of the methodological issues that come up with this sort of recent time demographic inference. So there seems to be, maybe this is suggesting recovery, but I'm going to put that on the side for now. But one of the other things you might be able to answer or explore with this data is whether there's important spatial variation. And when we first started to look at this, we saw a weird pattern just in a genome-wide PCA of these 3 super distinct clusters. And through a bunch of cool analyses, which I don't have time for today, we're able to determine that membership in these clusters is determined by inversion genotype at a single chromosomal inversion, about 31 megabases in size, segregating in these populations. It corresponds perfectly. And then once we mask that inversion from this PCA, the structure largely disappears. Right? A little bit about this inversion. It shows a really strong north-south cline, being more common in the north. To illustrate this point, really hammer it home, just fitting a linear cline to the inversion itself and genome-wide SNPs. The slope, which is demonstrated by the dotted red line, is quite steep. If you want to see it in a more formal GWAS framework, it's a very clear, significant association with latitude here, right? So seeing this, of course, begs the questions, especially if you've seen all the other great inversion talks this week, whether this is under selection, I believe it is, and whether this is involved in local adaptation. That's a developing story, as you might guess. We're still developing a lot of the genomic resources and otherwise to figure out what this inversion might be doing, right? But for the time being, it's something that might need to be considered in management given how striking this pattern is and that genotypes do very spatially. Maybe it's something to think about in the future. So what about outside the inversion? I showed you this PCA of the genome minus that, right? If we want to look at this more formally, we can look at isolation by distance. In this case, the x-axis is not plain old distance, but distance that a larvae might experience, right? So likelihood of dispersal from one site to the next, with less connected sites being towards me, more connected sites being that way, and plain old FST is on the y-axis. And we see a real flat line here, just showing that these less connected sites don't necessarily have higher FST values, so low IBD. Similar vein, we don't see much evidence for isolation by environment, or IBE. In this case, environment is a composite of a bunch of variables that intertidal black abalone might care about, things like air temperature, water temperature, et cetera. Right. So thus far, the takeaways I want you to take away are this high genetic diversity that's, again, kind of uniform across the range, and that this diversity is shared. We don't see much spatial structure with one super glaring exception. And I generally would like to think this is encouraging for recovery prospects, particularly because translocation from healthy populations is going to be likely the strategy people pursue. And concerns like outbreeding depression, you can maybe not weight as heavily given this preliminary evidence. But what about withering syndrome? So with our COVID swabs, we can also get diseases. A quick refresher, this withering syndrome is caused by bacteria. One thing I didn't touch on earlier is that it has been observed also with a phage hyperparasite inside the bacteria. And there's maybe some tenuous evidence that presence of the phage hyperparasite is linked to host disease severity, but jury's still out. What's cool though is we can detect both these things in our swabs, probably under detecting, as you might guess. We see both the phage and the bacteria in roughly 10% of samples, not perfectly overlapping, which I think is further evidence that there's probably ones we're missing here, right? If you observe the bacteria, or if you observe the phage, you would think its host is there too. But I think the more important takeaway is that given that sampling effort was pretty equal across the range, the presence of either the bacteria or the phage shows no significant association with latitude, right? So even if we're underestimating, it would still seem like the disease vector is everywhere across the range. Again, even though it's more common in the south, or the disease collapse is more common in the south. And same goes for this phage hyperparasite. So another quick summary. I touched on these points earlier, but I'm just gonna fast forward a bit. I think what's really exciting is with this sampling method, we were also able to look at the primary agent of decline in this species and show that, get a sense of where it is and how that might impact recovery strategies. And generally, I wanna say this is good news. My intent here is not to say that abalone are fine and they're gonna recover. It's that if the other pieces fall into place, like the management, all the ecological knowledge, all that stuff falls into place, the genomic considerations, which are what I'm bringing here, seem to indicate good things, right? And so some next steps, this is the thing that I am maybe most excited about right now. I'm in an ancient DNA lab, and we're right now getting pretty high quality genomes from shells. So the idea is to do a temporal comparison, particularly pre and post disease bottleneck, but then also deeper through time. We're able to get relatively affordable whole genome coverage from shells up to a couple millennia old. And if you want to talk about shell genetics and how this happens and discuss more later, I'm happy to do that along with any of the other topics. I'm actually pretty good on time. So yeah, with that, I just want to thank, this project was super collaborative. It was, the sampling was funded by the California Conservation Genomics Project, of which some of you may be familiar with, and there was a lot of cool genetics, ecology people, everyone associated with this. And then thanks to the abalone themselves too. And then with that, yeah, reach out to me and I'm happy to take what questions I can now. Yeah, Adam.
Okay, great. Hi, I'm Terbot. I'm a postdoc in Jeff Jensen's lab, and I'd like to talk a little bit today about the demographic history of the II Daubentonia madagascarensis. So some of you are probably familiar with the II, but in case you aren't, it's our charming little freak there on the right of the screen. It's one of the lemurs of Madagascar, so again, you're probably aware, lemurs are a group of primates endemic to the island of Madagascar. It's the 4th largest island in the world, and it's east of Africa. It also happens to be the largest nocturnal primate, and it's an insectivore with a similar strategy to woodpeckers, so it uses its incisors to dig into trees, to find different wood-boring insects, and then it uses a really kind of freaky, long, narrow finger to scoop those insects out and eat them. So obviously it's charming, so why would we want to study the demographic history? So for me, one of the interesting things is as a lemur in the group of strepsirrhine primates. It's one of the early diverging members of the primate lineage. And so if we're wanting to study different aspects of primate evolution, it's good to have an outgroup, and any sort of lemur, especially the II, makes a great choice. It's also of conservation interest because like pretty much all lemurs, possibly all lemurs, it's pretty endangered and threatened. It has various impacts from human interaction, so there's lots of issues with different habitat fragmentation with the forests that the II in Madagascar. And then, as we saw, it's the most charismatic primate, so it's another reason to study it. So why would we care about the demographic history of the II? Well, firstly, it's demographic history can affect the genetic diversity of an organism. So on the right, we have your classic sort of expanding population on the left, where you would get an accumulation of surplus of singleton alleles, while in a bottleneck or contracting population, you would end up with a sort of surplus of more common alleles. And that can obviously impact, if you're not accounting for this, if you're wanting to do something like study mutation rates and recombination rates for a lineage, or do genomic scans for evidence of selection. As well, if we're interested in the conservation of IIs, and we all should be, understanding its demographic history, estimating population sizes is obviously a very important part of conservation work. So next question we may have is, well, how are we going to estimate the demographic history of the IIs? So we're going to focus on neutral markers. So neutral markers are obviously going to be hopefully devoid of any sort of selective impacts on the allele frequencies. Specifically, if we're looking at neutral markers that are sufficient, about 10 kilobases from any sort of genic or selected region. And then we're going to use a sort of suite of different software to kind of estimate different demographic traits. So fast structure is really great for just getting the number of demes. So if there's any sort of subpopulations that we need to be considering. MSMC2 is really handy because you only really need to give it the number of demes, and then it'll kind of give you its best estimated model after that. And then the next software that we used is FastSimCol2, which requires a parameterization of intentional models. So for that, you have to tell it, I want to split the demes here, I want them to have a bottleneck or expansion, all those things. So we're kind of building up first, trying to find out how many demes there are, general idea of the demographic history, and then queue in with FastSimCol. So for our data, we have 12 genomes from a previous study. And now with this data set, we have 5 new samples, and the really interesting and nice thing about these new samples is this is accompanied by a new genome assembly. It has a lot higher coverage, and because of that, we're able to annotate the genes. And as mentioned, that's really important for determining neutral, non-selected regions versus those genic regions. We're also excluding repeats because they're less informative. It's just repeating sequences over and over. And then also there's sets of constrained regions among primates that across all primates are relatively constrained. Since they're constrained, that implies there's probably some sort of heavy background selection on those regions, and so we're also wanting to exclude those. After that, we still end up with 126 million, a little over, sites across the 14 autosomal chromosomes to look at. So our first sort of parse, again, is fast structure, and from that we get a pretty clear signal that there's a strong sort of just 2D model. So when we just test for 2, we get a clean split between samples from the northern area part of the range and then everything else. And then when we test it with a model for 3 demes, it comes back and says, no, you really want 2 demes. So technically, there's some membership into the spurious third deme, but it's very small on all of the samples, so small that you can't even see the third color on that sort of structure plot. So we have pretty good confidence that we have 2 demes that we're dealing with, split in this northern area, and then everything else. So when we put that into MSMC2 and try and get our results back, we get something we might expect with something that's of conservation interest, where we have a bottleneck far in the past, and then maybe some recovery currently. A few kind of questionable things. It's expecting this bottleneck to have occurred about 10,000 generations in the past. The generation time of an II is about 5 to 7 years, so that would put it 50,000 years in the past, which doesn't necessarily correlate with a lot of known things, so it's a little suspicious. And then the population sizes that it's estimating are, in the hundreds of thousands, including after the recovery, which also seems a little suspicious. But from this, we can say we should probably be testing models that involve some sort of population decline and possibly an expansion more recently. And so with FastSimCool, we did exactly that. So we sort of tested 3 sort of overarching models. So we have one where it's just a population split from an ancestor into 2 different demes, and then just carried through time. We have one where there's a population split and then a continuous decline from that split. And then we have one where there's 2 demes with that population split and then recent growth. It's important to note that the way FastSimCool works, the sort of ratio between the sizes of those 2 demes when we're testing this is not a priori heavily prioritized. You give it some range, but it will still be able to go outside that range. So it's going to test, is this deme larger than that, across that, as well as the timing of the bottlenecks and the timing of the growth. We also tested sort of 3 subsets of these models, with one without any migration, one with sort of symmetrical migration, and then one with an asymmetrical migration. So we then ran these models, looked up, and we get our maximum likelihood scores. And so we can do some comparisons there, but what we really want to focus in is our AICRI Keiki information criterion, which is just a adjustment for maximum likelihood that accounts for the number of parameters. So if you have more parameters in your model, it's going to penalize it. And we're looking for the lowest AIC. And for that, it's pretty strongly the model with 2 demes, a bottleneck, and then some recovery with no migration at all. Generally in AIC, you're looking for, to say these models are, maybe both good, you're looking for ranges of less than 100 in your delta AIC, and we really only have the bottleneck without recovery that falls in that range. The next one is, even further behind, and that's just the decline. So at least from these results, there doesn't seem to be a lot of migration between our 2 demes. To confirm these results, we then took the best parameter model combinations to use as model parameters, and then ran those as simulations to generate simulated SFSs to just be able to visually compare the simulated site frequency spectrum to the observed site frequency spectrum. And so the site frequency spectrum, if you're unaware, it's just a histogram of the allele counts in a population. So is there just one of these alleles, 2 of these alleles, et cetera? And so if we look at these, here's our best performing model. We have the population split, bottleneck, and recent growth. And the SFSs between simulated and observed are actually quite close. They're very close, which is good. It's a good sanity check there. And so this specific model has parameters where the effective population size of the ancestral group is about 23,000 individuals. That occurred about 1,000 generations in the past. Recovery growth only was occurring about 10 generations in the past. Pretty slow, but still a little bit. And then our current population sizes, the North Deme is about 3,000 individuals, effective population size, and everything else is about 7,000. If we look at our other models, so these are the ones that are performing a little less, you can see this is still pretty close, but the difference between our 2 sets of bars, are starting to spread apart. We're getting similar sorts of stories from just the bottleneck-only model, where the ancestral deme is about 23,000 individuals, with the bottleneck about 1,000, and again, population size is comparable. So it's nice that the next best performing model is giving us similar parameterizations, but it's lacking that growth, which seems like it's important, which is good when we see that endangered species has signs of growth in its population size. And then last, our population decline. It's a mess. So it's not surprising that this was, the 3rd but best performing model, but it's still far behind. Our SFSs are pretty far apart. The ancestral population size and the time that it started are still matching, as well as sort of our size, but we can probably say that the decline in population was not a gradual, over time, II populations are decreasing, but there was some sort of event that caused perhaps a drastic reduction, at least in the terms of, II generation. So over the course of a few decades, a couple decades, it went from their ancestral population to a much smaller one. Uh, there's supposed to be a picture here of the illustrating the decline in forest cover, but it didn't upload correctly, so if you're wondering why there's a big empty space, that's that. And then just a reminder, that's sort of what our model, our best performing model looks like. And just to note, those are to scale, so visually this is a relatively accurate depiction of what's going on. So our current effective population size is quite low. It's about 5,000 to 7,000 individuals. Diploid, right. Diploid individuals, so the effective population sizes that we mentioned before are in terms of haploid individuals. So other note is these are effective population sizes, not census population sizes. So 5,000 seems a little scary, but that just means their diversity is low. There could be more than 5,000 individuals out there, thankfully. We have the population decline occurring about 1,000 years ago. This is most likely sudden. This corresponds approximately with the arrival of humans to the island about 3,000 to 5,000 years ago. We know that when humans tend to come to a place, they can disrupt ecosystems, especially, other mammals and birds and large things that we probably are eating or doing other things to. So that kind of is a possible reason for the bottleneck. And then we have this slow recovery about 10 generations prior, and that corresponds to the rediscovery of the II and the initial move to do conservation measures. So we have this sort of promising sign that conservation is at least working a little bit. For our future directions, we want to look at some models with deemed-specific timing and rates for growth, just to see if we can fine-tune those differences between the observed and simulated SFSs and get those to match a little closer. And then we are going to use this demography, specifically the other, since that's where our 5 high coverage genomes are from. And with that, we're going to be able to use the demography to account for diversity variation to do things like selection scans and population estimations of mutation and recombination rates. And with that, I'll just acknowledge the Jensen and Pfeiffer labs who have helped me a ton with this, funding sources, and then the computing resources with Arizona State and the OSG pool. And I maybe have time for one question or two.